The virus that causes COVID 19 today is not the same as the one that first sickened people way back in December 2019. Many of the variants circulating now are partially resistant to some of the antibody based therapeutics that were developed based on the original virus. As the pandemic continues, more variants inevitably will arise, and the problem of resistance will only grow. Researchers at Washington University School of Medicine in St. Louis have identified an antibody that is highly protective at low doses against a wide range of viral variants. Moreover, the antibody attaches to a part of the virus that differs little across the variants, meaning that it is unlikely for resistance to arise at this spot. The findings available online in the journal Immunity could be a step toward developing new antibody-based therapies that are less likely to lose their potency as the virus mutates. The researcher says that the current antibodies may work against some, but not all variants. The virus will likely continue to evolve over time and space. Having broadly neutralizing, effective antibodies that work individually and can be paired to make new combinations will likely prevent resistance. SARS-CoV-2, the virus that causes COVID-19, uses a protein called spike to attach to and invade cells in the body's respiratory tract. Antibodies that prevent spike from attaching to cells neutralize this virus and prevent disease. Many variants have occurred mutations in their spike genes that allow them to evade some antibodies generated against the original strain, undermining the effectiveness of antibody-based therapeutics. To find neutralizing antibodies that work against a wide range of variants, the researchers began by immunizing mice with a key part of the spike protein known as the receptor binding domain. Then they extracted antibody-producing cells and obtained 43 antibodies from them that recognized the receptor binding domain. The researchers screened the 43 antibodies by measuring how well they prevented the original variant of SARS-CoV-2 from infecting cells in a dish. 9 of the most potent neutralizing antibodies were then tested in mice to see whether they could protect animals infected with the original SARS-CoV-2 from disease. Multiple antibodies passed both tests with varying degrees of potency. The researchers selected the 2 antibodies that were most effective at protecting mice from disease and tested them against a panel of viral variants. The panel comprised viruses with spike proteins representing all 4 variants of concern: alpha, beta, gamma, and delta, 2 variants of interest, kappa and iota, and several unnamed variants that are being monitored as potential threats. One antibody, SARS-238, easily neutralized all the variants. Moreover, a humanized version of SARS-238 protected mice against a disease caused by 2 variants, kappa and a virus containing the spike protein from the beta variant. The beta variant is notoriously resistant to antibodies, so its inability to resist SARS-238 is particularly remarkable, the researchers noted. Further experiments pinpointed the precise spot on the spike protein recognized by the antibody and identified 2 mutations at that spot that could, in principle, prevent the antibody from working. These mutations are vanishingly rare in the real world, however. The researchers searched a database of nearly 800,000 SARS-CoV-2 sequences and found escape mutations in only 0.04% of them. The researcher says that this antibody is both highly neutralizing, meaning it works very well at low concentrations, and broadly neutralizing, meaning it works against all variants. That is an unusual and very desirable combination for an antibody. Also, it binds to a unique spot on the spike protein that isn't targeted by other antibodies under development. That is great for combination therapy.